Hello and welcome to the screencast on excitation contraction coupling. A couple of terms we want to define first. Simply, excitation means stimulation by a motor neuron, and you are going to see that this stimulation causes an electrical event or a change in the membrane potential. And then contraction is the physical interaction between myosin and actin. So you remember this is myosin grabbing onto actin and pulling. So we've got this electrical event that takes place at the cell surface called excitation, and then this physical event that takes place in the cytoplasm called contraction. And we have to figure out how one causes the other. How is excitation coupled to contraction? So this is a brief outline of the steps of excitation contraction coupling. This is not the complete answer to the essay question on the exam, so make sure you can fill in the gaps in this outline as we go. But this might be just a handy way to think about it, to start with just listing the steps. And what you want to remember is that steps 1 through 3, all of this is just electrical signals. So even if the terminology is confusing and new, you can still just sort of think of it like electricity being conducted from point A to point B. It's a little bit of an oversimplification, but if you think about it that way, you're used to thinking about electricity running through wires. Now, instead of running through wires, it's running through parts of a cell. So it is going to start with the end plate potential. If you recall, the motor neurons use this neurotransmitter called acetylcholine. When they get stimulated by your central nervous system, acetylcholine is released into the synaptic cleft, or the space in between your neuron and your motor end plate. This neurotransmitter is going to bind to the neurotransmitter receptor, or the acetylcholine receptor, which we said is a ligand-gated ion channel. So the receptor pops open, sodium rushes in, this depolarizes the motor end plate and causes an electrical phenomenon that we refer to as the end plate potential. So that was a lot of explanation. Concentrate on what's listed here. It should be very similar to what was in the original PowerPoints. I tried to clean up the language a little bit to make each step a little bit more succinct. So acetylcholine gets released, it binds to the receptors, depolarizes the cell or the motor end plate. This causes an end plate potential. And that was this part right here. So it's depolarization. The depolarization of the motor end plate is going to trigger a depolarization of the rest of the sarcolemma. This is referred to as the action potential. So one electrical event here is going to trigger another electrical event here, and this current is going to flow across the surface of our cell, and we're calling this the action potential. This action potential, as we said before, travels across the surface of the sarcolemma, and because the T-tubules are just extensions of the sarcolemma, the action potential also travels down the T-tubules. These T-tubules are right up against the ends of the sarcoplasmic reticulum, and when the action potential gets down to where the sarcoplasmic reticulum is, it triggers the release of calcium. So here you have some electrically sensitive proteins that are coupling the T-tubule with the sarcoplasmic reticulum, and when this electrical signal travels down the T-tubule, it delivers a little shock to the sarcoplasmic reticulum, and then the calcium is no longer stored just in here, but it's free to float around in the cytoplasm and to come in contact with our actin and myosin. So the presence of the calcium in the cytoplasm is going to trigger a contraction, which is the cross-bridge cycle that we talked about in lecture. And that is going to continue until the calcium is pumped back into the sarcoplasmic reticulum. So that is it. 6 steps as succinct as possible in under 5 minutes. Way to go, me. Thanks for listening.